Do you know that the true VIPs of nutrition can be found at the produce section of your grocery store? In this lecture, Dr. Jed Fahye will bring us on a fascinating journey into the world of very important phytochemicals, the VIPs of healthy eating. You'll discover the superstar nutrient sulforaphane in broccoli and other cruciferous vegetables. Plus, you'll learn about some kitchen chemistry tricks to cook your veggies and maximize their sulforaphane content. Get ready to supercharge your diet and bring your health to the next level. I am Dr. Lucia Aronica, and I am on a mission to make the science I teach in the classroom accessible to everybody. If you want to support this mission, please make sure to subscribe to my YouTube channel and hit the notification bell if you want to get notified when I post a new video. So, let's jump into it.
At the time that sulforaphane was discovered in the early 90s, all of the media started talking about, it's in a cruciferous vegetable, broccoli. And the fact of the matter is there are other similar compounds in other cruciferous vegetables, but broccoli is pretty much the sole edible source of sulforaphane itself. For those of you who are not card-carrying botanists or who don't look at the labels when you go to the grocery store and just chuck stuff in your cart, these are some of the cruciferous vegetables. They all belong to the same family. They all have similar compounds to sulforaphane, which we'll talk about a lot in a few minutes. But arugula, the Chinese cabbage, collards, turnip, canola, which is most of you are familiar with as an oilseed crop, rutabaga, mustard greens, radish, nasturtium. These top and bottom aren't really that relevant as an item of culinary interest in this country. And then, of course, cauliflower, cabbage, Brussels sprouts, and broccoli. And I'm showing you broccoli sprouts there, which we'll also talk more about. So I was growing broccoli on 15 or so farms on the eastern shore of Maryland, various locations. We were screening a number of broccoli varieties and stages of growth for sulforaphane levels. And then when the winter came, I couldn't get broccoli from the field anymore. And I was on the 13th floor of the Johns Hopkins Hospital, main hospital in the middle of East Baltimore. There are no rooftop gardens there. There weren't and probably still aren't. So I had to use an incubator and start getting smaller and smaller. And being trained as a plant tissue culturist a long time ago, I started taking broccoli seeds, surface sterilizing them, meaning getting rid of the contaminating bacteria, and growing sprouts in an incubator. Make a long story short, we found very high levels of the precursor of sulforaphane, which I'll tell you about in a minute, in broccoli sprouts. 3-day-old sprouts is what we were using. It turns out that the levels of glucoraphanin, the precursor of sulforaphane, were even higher. They were the highest in seeds. But at the time, Paul and I were thinking that we wanted to do something that would help people and their diet and their selection of vegetables. And nobody was eating broccoli seeds. Nobody still does, and nor really should they, for reasons we can talk about later. But there were, a handful of people in the country that ate green sprouts. And, it's a very small number. I don't know if it's 1% or less. And we thought, well, sprouts are green. Some people should be interested in eating broccoli sprouts. And so we actually worked on broccoli sprouts for a long time. And in a sense, I'm still working on them. Just to finish this graph out, when broccoli heads mature, for those of you who don't know how seeds are formed, each of these little buds on the broccoli head turns into a seed pod, and then those seeds are harvested a couple of months later, and you get these. So let me not dwell on that too long.
I have a question, if I just want to show now. So you mentioned that broccoli seeds are the richest source of glucoraphanin, which is the precursors of sulforaphane, but that we shouldn't be eating them. I think that the seeds or seed powder is an ingredient in many sulforaphane supplements on the market. I may be wrong. I don't know whether you can comment on that.
Of course, yeah. So I'll make a very long story as short as possible. So the seeds are bitter. The raw seeds are bitter. They taste a little bit nutty if you bake them, but the window of time you have to bake them for is very small between baking and burning. They're about 30% oil. And that oil contains some erucic acid. Lorenzo's oil contains some oil and other compounds that maybe not advantageous to people to eat a lot of, besides the fact that there's a lot of fat in these seeds or oil. So from a calorie perspective, if you were to eat a lot of them, that might not be particularly wise. Paul and I started a company in the mid or the late 90s, which still exists today. And the company was put on the map to promote and commercialize broccoli sprouts. There were FDA alerts about safety of all sprouts. There were never any outbreaks in broccoli sprouts, but the commotion that was raised about sprouts in general was enough to turn a lot of people off, including some potential business partners of ours, to sprouts, to promoting them, to the potential risk of them. So the company turned into a company that makes an ingredient for supplements. And to your point, Lucia, they make this ingredient from the seeds, again, the richest source of glucoraphanin, but it's an extract. It's a water soluble extract. So they leave all the fats behind and essentially concentrate the glucoraphanin into a powder that is sold by them. The company's name is Brassica Protection Products, and make no secret of it. And they sell this BSE or broccoli seed extract to supplement companies that make many of the supplements you're talking about, I think. So it's not actually a seed powder, it's a seed extract powder.
I see.
Yeah.
Because I have, so this is a personal question, because I started to just experiment with having a teaspoon of broccoli seeds a day, just chewing them. I wonder whether, what do you think about this habit? I mean, is this too much? Is this enough to give me enough sulforaphane? I know that there are some anti-nutrients in seeds. Yeah. I'm just thinking that probably 1 teaspoon may be the sweet spot to have benefits without too many negatives. I don't know. What do you think about it?
I think you've hit the sweet spot exactly. A teaspoon is what, about 5 to 8 grams of seeds. I think they're bitter and not good tasting. So I would put them in the oven at 350 in a tray, single layer in a tray, and you really have to watch them. I mean, something like 3 to 5 minutes. If they start to scorch, they're going to taste really bitter and nasty. If you catch them at the right point, they'll taste nutty. And you will have killed. Yeah, and you will have not killed. There's some scientists here, I can't use the word kill, talking about an enzyme. You will have denatured and inactivated the enzyme that's responsible for, this is actually a good setup for my next slide, for converting this glucoraphanin precursor, which is what is in the seeds and the sprouts, to sulforaphane. Because enzymes don't like to be cooked. You know, for those of you, again, non-scientists, envision frying an egg. All the white of the egg is protein, and it's clear and vitreous. And when you put it in a frying pan, it turns white. That's because those protein molecules are denaturing irreversibly. They don't go backwards once you denature a protein usually. So what happens with this phytochemical system, which has been dubbed the mustard oil bomb because these compounds are called known as mustard oils. There's an enzyme called myrosinase present in the same tissues as the precursor. The precursor, glucoraphanin, is water soluble. It's typically found in vacuoles of the plant cell. It's heat stable, and it's very non-reactive. It doesn't do squat for us as human beings. It doesn't do anything just as it sits. The enzyme, myrosinase, acts very quickly, and it converts glucoraphanin with an intermediate step to sulforaphane. Again, these are, well, not again, I didn't tell you this yet, but these are all called glucosinolates, and these are all called isothiocyanates, molecules that all have an N double bond C double bond S tail on them. And there are about 130 of them in plants, almost all in cruciferous vegetables. And it's this part of the molecule that varies. So 130 different ends or tails or on these molecules. Sulforaphane is reactive in our bodies. It's heat sensitive, so it degrades very quickly, and it's not as water soluble as glucoraphanin. So what happens is when you homogenize a plant that contains these precursors, you allow the membrane-bound enzyme to come in contact with the floating, dissolved glucoraphanin. When you break a cell, these two things come together and you make this reaction. It's easy to envision, an insect that the plant doesn't want to get consumed by, taking a bite out of a plant leaf, and this, it releases these myrosinase and glucoraphanin. They come into contact. They form this compound sulforaphane, which is a feeding aversive compound, feeding deterrent for many insects. So they go away. They prey on something else. Same thing with bacteria in the plants, bacterial infection or fungal infection. Those infections involve breaking cells. Those liberate these compounds and cause them to happen. So when you ingest, when you chew or masticate a raw cruciferous vegetable, you're a predator. You're a mammalian predator, and the plant is doing the same reaction in your mouth and as it goes down into your gut. It's more difficult to sort of visualize that with broccoli, but a very closely related plant is the red radish or wasabi or watercress or arugula. And most of you probably realize when you chew on something like that, or a mustard seed for that matter. But let's take red radish for an example. First sensation is cool and crunchy. Then within 20 or 30 seconds, it's hot or very hot. That's because the analogous compound to sulforaphane, very closely related, similar NCS group, that molecule is very hot. It's a lacrimator. It makes you cry, makes your nose run. So that's the reaction that then
very interesting. I didn't know that. I introduced the topic of this reaction in this class, and I used the analogy of a glow stick, that when we break it, we produce light because we mix two components that are usually separated, and then when they come together, we have a light reaction. And I have a question also on this slide. So
yes.
Basically, yes, glucoraphanin is an example of glucosinolate, and there are other glucosinolates in other cruciferous vegetables. And then sulforaphane is the active epi-nutrient or phytochemical that is good for us. Now, we need to be careful when cooking. For example, we can degrade myrosinase, and also sulforaphane is heat sensitive. My question to you is, I teach my students that by chopping their cruciferous vegetables 30 minutes before consumption, they can activate a little bit this reaction that you are showing here, so that some sulforaphane is produced, but then the point is they shouldn't be cooking broccoli a long time to prevent sulforaphane to be killed. And so another way of doing that could be fermentation. My question to you is that if we, for example, ferment cruciferous vegetables, cabbage, is the isothiocyanate that is produced by that reaction by chopping stable during the fermentation process or not?
So, good question. I'm going to choose to answer the first part of your question a little bit more thoroughly, but the short answer to the fermentation question is probably not, because most of these isothiocyanates, I mean, you'll get some, but most of these isothiocyanates are like to bind to other organic material. So if you put sulforaphane in a just dissolved pure sulforaphane in water, you can sit it out on the counter for days and it's stable because there's nothing organic for it to react with. If you put it in a mess that's created from grinding up the plant, or from grinding it or cutting and fermenting it, it's going to bind to organic plant matter fairly soon, fairly quickly. But not all is lost. And I should say that one of the things that I think we all have to be cognizant of is that getting people to change their dietary habits is incredibly difficult anyway. I mean, just without talking about what vegetables they should be eating, right? And it's incredibly difficult, and it's incredibly difficult and notoriously difficult to do dietary surveys and to quantify or measure what people are really eating and how they're changing their diets. So, I mean, I think I've learned the hard way that, you've got to be careful making elaborate cures for a disease that people don't even want to accept they have, that disease being unhealthy aging. And so going to the extent of fermenting, for example, broccoli, when most people won't even eat broccoli, can be difficult. Now, you and I have Italian heritage, so we come from places where people do eat those kinds of vegetables and the Mediterranean diet and so on. But there's more, as they say. So on this, I think it's on this slide. Yeah. So what you're asking is, put a big X over the raw vegetable. What happens if you cook it? You brought up cooking, and when you cook it, you denature the enzyme. No question about it. So then we get to count on our gut microflora. And it turns out that all of us, every person on this call, every person in this class, we have not actually found any exceptions, but everybody who has a relatively even not that healthy, but a relatively healthy gut microbiome has bacteria in their gut, mostly the large intestine, that contain myrosinase activity. So that cooking broccoli and ingesting it with no myrosinase added still has benefit, because the bugs in your gut metabolize or convert glucoraphanin into sulforaphane. And there's actually even an argument to be made that it may even be better, or certainly as good for you, because it's producing it in right where it's going to get absorbed, as opposed to ingesting it in the oral cavity, in the esophagus, where it turns out there's really not much absorption of sulforaphane, because we and others have checked. So I mean, if you envision, let me stand up and do the sort of anthropomorphic thing, but if you envision the glucoraphanin is coming in here, and if sulforaphane is being released, either because you eat something rich in sulforaphane, or you deliver something that's converting it, a lot of that has the potential to be absorbed or inactivated and bound up in this part of the body. And so the kinetics are quite different than if you count on the gut microflora, which, are sort of here to here. And so that's why, in the title of the slide, I'd forgotten that's on the title, I put the slow release solution, because letting the gut microbiota or microflora do this catalysis, do this enzymatic conversion, is a much slower and more gentle delivery system, if you will.
And this is good news. And thank you for emphasizing this concept. I shared with my students the concept that our microbiome can help activate, produce sulforaphane. So basically, the good news is eating broccoli is anyway good for you. There are many ways that bring to Rome or to sulforaphane, right? You can chop it, chew it, and even if some myrosinase is inactivated through cooking, or if you lose some sulforaphane through fermentation, anyway, you will be able to extract some more sulforaphane through your microbiome. And so eating broccoli is a good idea, right?
It will all go to a good place, right? We thus call sulforaphane an indirect antioxidant. And I want to just take a minute and I think I'll stop with the hardcore sort of chemistry or chemical biology then. But when you generally hear people, especially people in the nutrition field, talk about antioxidants, they're talking about vitamin C or lycopene or something. Let's take vitamin C for an example, where you ingest it, it has direct antioxidant capacity, meaning it is oxidized by compounds you may not want in your system, and then it's done, it's finished. So every molecule that you take in of vitamin C, once it's done its thing, forget about it, it's gone. So, and in fact, it spikes in the blood very quickly and in the system, and unless you're ingesting it multiple times a day, I would argue that it may be of somewhat limited value. Certainly some stays in the blood, it has a half-life, et cetera, et cetera. But then contrast that to what I just showed you with sulforaphane and the NRF2 pathway. And this is an alphabet soup. You don't have to pay attention to it, it doesn't matter. But my point here that I'd like you to at least hopefully appreciate is that each of these yellow bars is a type of, a group of genes, a classification of genes. So antioxidant enzymes, carbohydrate metabolism, heme metabolism, biotransformation, et cetera. The alphabet soup is the actual enzymes. What I want to point out, though, is that the half-life of these enzymes, once upregulated, ranges from hours to days, many days in some cases. And what that means is that this simple molecule, this phytochemical or epi-nutrient, when you ingest it, it spikes in your blood just like vitamin C. The pharmacokinetics look almost identical to vitamin C. So it's and it goes away. You pee it out. We measure it in the urine all the time in clinical studies. But what it has done is it has upregulated, cranked up, boosted, if you will, this whole suite of enzymes that stay around and continue to do their thing. Most of those things, almost all of those things, very positive for your system for hours to days. So you don't really even have to eat broccoli sprouts, every day. You can eat them every 2 or 3 days to good effect. So one of the things that are upregulated is this molecule called glutathione. Glutathione is a tripeptide, 3 amino acids linked together. It's, I think, useful to know to any science class that it's the most abundant, low molecular weight antioxidant, not only in animals and in us, but in many bacteria and plant cells also. It occurs, for example, in neurons at very high levels to protect the neurons. And it regulates a whole suite of critical cellular processes. So it's a first-line defense mechanism that we have in our bodies. I think the highest levels are present in the macula of the eye, in the retina. It protects against photooxidative damage. I think I don't have any other mention here, but neurons in the eye are the things that really stand out. But in all cells of our body, really, this is our endogenous antioxidant. Let me just skip a couple of these. Levels of glutathione decline with age. This is the slide I wanted to get to. So the glutathione cycle, or the mechanism by which glutathione is regenerated, or the reduced, it toggles back and forth between its reduced and oxidized stage, involves a number of steps where the NRF2 pathway is critical. I mean, this is one of the rate-limiting enzymes in making glutathione, and it's potently regulated by NRF2. So you can enhance your glutathione status without resorting to things like eating N-acetylcysteine supplements or some of the other supplements. And certainly sulforaphane, in addition to having a direct effect on this enzyme, as we've discussed, upregulates NRF2. I mentioned N-acetylcysteine only because when I did a recent scan of the literature on dietary supplements for a paper we did a year or so ago, it turns out that N-acetylcysteine is the most talked about is that N-acetylcysteine and CBD from cannabis are the two compounds that have most clinical trial activity associated with them. But when you eat as a supplement N-acetylcysteine, it's digested. I mean, it's not much of it goes into feeding this pathway. So it's a very dubious distinction that there's so many clinical studies looking at it. So we and others now over the years have found very broad protection by sulforaphane in the clinic with people because NRF2, because glutathione, because all of these things are present in essentially all of the organs of the body. We and others now have been looking at it via the NRF2 pathway primarily for things like autism and Alzheimer's. I don't think there's any clinical evidence. There's some animal models. Parkinson's disease. There's a fascinating story on that. If someone asks me about it when I shut up, I'm happy to tell you about it, etc. So, as I say, a wide range of studies that have been done over an extremely wide range of doses. Most of the studies have been done with, on this bar on the bottom, healthy individuals. That's frequently the way one starts doing clinical studies where one's looking at safety. And it's not a drug, it's a food that can be delivered in a supplement. But there have been a substantial number of studies looking at specific conditions. For example, Helicobacter pylori infection. I did a study with colleagues in Japan on that and a number of years ago. Well, thank you for having me. And I'm sorry. Yeah, there are a lot of slides talking about specific.
We can have you another time in this course, and I think the topics you touch are really at the core of the course. I also think that the NRF2 pathway you mentioned. So, just for the students that are not familiar with that, there are many ways of activating gene expression. So, epigenetics is defined as a stable modification of either the DNA or the histones that regulates gene expression, right? And then there are these transcription factors like NRF2 that can open and close the chromatin by regulating so-called chromatin remodeling enzymes. But some of these two mechanisms are overlapping. So I'm sure that part of the NRF2 mediated activation of gene expression is also involving some epigenetic mechanisms that are inherited through cell division. So that's So thank you very much.